Director at BLD University. Welcome to you, sir. And we have our KSTA esteemed member, HS Savitri Madam. Welcome to you, Madam. And before beginning the talk on chemistry Nobel Prize for protein design, Professor M.R.N. Murthy, sir, just a brief overview of his achievements. Professor has done his BSc from Bangalore University in 1970, then pursued his MSc from IIT Madras in physics and obtained first rank in 1972, and then pursued his PhD from the very renowned Indian Institute of Science, Bangalore, on X-ray crystallography in 1977. Sir started his illustrious career as senior research fellow in 1982, then became assistant professor, then he became professor in 1994. Sir is also INSA senior scientist. Then from 2018, Sir has been AstraZeneca distinguished professor at DAB Bangalore, and 2019, Sir has been a visiting professor at IISr Thiruvananthapuram. His research interests include virus structure and assembly, macromolecular crystallography, protein structure, function and evolution, X-ray diffraction. Sir has received numerous awards from different institutions and Government of India. He was adjunct professor at Purdue University from 89 to 92. He was elected to the Indian Academy of Sciences in 1992. Sir has been the recipient of a very distinguished scientific award, the highest award in India, Shanti Swarup Bhatnagar Prize in 1993, and he is also elected to the Indian National Science Academy 1996, and the Indian Science Platinum Jubilee Lecture Sir delivered in 1990. And he is also a fellow of National Academy of Sciences in 2001, and Rustum Choksi Award for Excellence in Science in 2022 he received. Sir also has received R.L. Kapoor Endowment delivered lecture and Ramanujan by Mathematical Society in 2003. And Sir has received G.N. Ramchandran Memorial Award in 2003. And he is also a fellow of the Third World Academy of Sciences, then AstraZeneca Distinguished Scientist, UGC Hari Om Ashram Trust Award. He is received in 2004, Jagdish Chandra Bose Award for Life Sciences. Then many more such awards Sir has received. With this very brief introduction, I request Murthy Sir to take over and deliver his talk on Chemistry Nobel Prize for Protein Design 2024. Now over to you, sir.
Thank you very much. I am extremely sorry that this delay occurred. I apologize. I also want to thank the office bearers of Karnataka Science Academy for asking me to give this lecture. It is an honor and pleasure. And it is also a very great pleasure to present some results that are related to my own field of research. I presume that the audience consists of not only students of science, but also people from other disciplines. So I would like to make this seminar accessible to all audience, which means it will be essentially somewhat elementary. And if there are experts in the audience, please bear with me. Just to put it in a nutshell, the reason why David Baker was awarded the Nobel Prize is that if you look at the screen, on the left side, you see a molecule. This is a very complex molecule, consists of hundreds of atoms. How to make this molecule, how to synthesize such a molecule or create such a molecule is a challenge to any chemist. You know, the amount that will be spent to make such a molecule will be phenomenally high. And David Baker made it. And on the right side is the structure actually determined experimentally of the molecule that was made by his team. And you can see the similarity between left and right side. So whatever the molecule they made, with a view of giving it a particular structure That is precisely the structure the molecule had when experimentally observed. This is the greatest achievement. It is a great achievement in chemistry, great achievement in biology, great achievement in pharmaceuticals, as you will see later in the seminar. For this, he was awarded the Nobel Prize. So, in a nutshell, we know why he was awarded the Nobel Prize. We will now get into some nitty gritty details. You know, if you look at the surface of Earth, which teems with living organisms, you will see the molecules on the right. You take a soil anywhere on the surface of Earth, you will find in that soil proteins, nucleic acids, lipids, and carbohydrates. These are relatively large molecules. You will also find smaller molecules such as amino acids, nucleotides, fatty acids, and sugars. These small molecules are not very specific to Earth. You know, people have found remnants of these molecules in asteroids. Asteroids may have carbon, water, and nitrogenous salts, and such asteroids could have these small molecules. It is not surprising to find these small molecules in exoplanets, that is, the planets circling around other stars. But proteins and nucleic acids you will never find where there is no life. So they are actually molecules of life. They are the hallmark of the fact that life is present in that place. So these molecules are abundant on Earth. How have these come about? What kind of speciality these molecules have is a very fundamental question in biology, biochemistry, and we will look into these things. To summarize this slide, the small molecules are very common in the universe, the large molecules are exceptionally rare. Well, only life makes it. So, what is the secret of this life? You know, how do living molecules, living cells manage to synthesize these molecules? The code is very well known now. It is known for about 70 years. We have in our cells a molecule which all of you have heard, deoxyribonucleic acid, that somehow has appeared on Earth, and that has information for the synthesis of proteins. That means proteins are made using the information on the DNA. And how this molecule proteins are made using nucleic acids is very well known. But can we make new proteins using some nucleic acid which doesn't exist in living cells? Is a question which is very interesting. We will see the answer to this in the later slides. Anyway, all our genetic information, the fact that we get angry or we can learn language, we can smile, all of this is the result of our DNA. And that the DNA is expressed in terms of proteins. Without proteins, the information on DNA is dead. If that information has to be expressed, it has to first get translated into the form of proteins, and proteins express. How do proteins express? First of all, proteins make rigid parts of our body. The skin that we have is a protein, and the skin protects all internal organs by spilling into the natural environment. So that is a protein. Similarly, the cells have their shape because of proteins. Proteins also carry out reactions. It is a wonder how proteins carry out reactions. For example, the sugar stored in your kitchen will stay as sugar for an year or many years, but you eat a spoon of sugar, in a few hours, it won't be in your body. It will be probably burned into carbon dioxide and water, and you will get some energy. So, reactions that take place in the body are millions of times faster than the reactions that take place outside the body. That is why you see athletes are able to run. When they run, so many chemical reactions have to take place in their body. If they are slow, no action is possible. And proteins make this fast action possible. So proteins are the workhorses of our cells, whereas DNA is the genetic material. If there is any change in the DNA, there will be change in the genetic material. Now, let us look a little closely at what these proteins are. Here is a very colorful bead, a colorful chain of beads. Proteins are similar. They are chains of beads, except that the beads are not plastic pieces, but they are amino acids. Amino acids are small molecules, as I pointed out in the earlier slide. They are available in probably exoplanets and some comets. There are life users. You see, actually, there are virtually hundreds of amino acids that a chemist can synthesize. Since these are small molecules, a chemist can actually synthesize amino acids. But life uses essentially 20 amino acids. It is not strictly true, but for our purpose, as of now, we can take that life uses 20 amino acids. These 20 amino acids and their structures are listed on the right side diagram, and their names are also given. But you don't have to really look at all the details, but I will just show you the color code here. The color code here, you see, there is red here. The red color coded amino acids have a negative net charge on them. You know, the universe is full of positive and negative charges. Our body as a whole has no net charge because the amount of positive charge is exactly equal to the amount of negative charge. But if you take these individual amino acids, as they occur in most of the times in living organisms, they will have a negative charge. Whereas one on the right, shown in blue, are all positively charged. Whereas the amino acids in green, they can make the so-called hydrogen bonds with water. We will look at hydrogen bonds in a minute, but they are very essential parts of protein structure. So these can make hydrogen bonds. The one at the top, the amino acids in gray, they are hydrophobic. Hydrophobic means they tend to avoid water. They are more soluble. Actually, they are less soluble in water compared to other amino acids. They like to get away from water. You see, it is just like butter clumping when you churn buttermilk. These amino acids tend to avoid water, whereas all other amino acids, other than one shown in the top, would like to interact with water. They want to be in the presence of water. So proteins are really chains made up of amino acid beads, and there are 20 different beads. But then if you take, for example, a protein of 200 amino acids length, the reason to choose 200 amino acids is that it represents typically the size of proteins in our body. So if you take 200 amino acid, for the first amino acid, you can use any one of the 20 amino acids. For the second position, you can use any one of the 20 amino acids. Like that, for all the 200 positions, you can use any one of the 20 amino acids. So simple statistics will tell you that the number of possible different chains is 20 power 200. If this is same as thinking, if you have 200 beads in the previous slide, you know, the plastic beads, and you have 20 colored beads, how many different necklaces, open necklaces you can make if it is 200 beads? Since you can use any colored bead for any position, the number comes out to be 2 power 200. 2 power 200, if you raise it to 10 power instead of 20, base instead of 20, you change to 10, it becomes 10 power 260. That means 1 followed by 260 zeros. What is this giant number? None of us can really imagine. You know, we can only say this is very, 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 very big. 1 followed by 260 zeros. For example, just to give a perspective of what we are talking about, the average number of stars in our galaxy is 10 power 8. 10 power 8 is 1/10 of a billion or 100 million. Estimated number of stars in the whole universe is about 10 power 23, or take it as 24, then it will be a million, 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 or a billion, billion stars. Estimated number of atoms in our whole universe is supposed to be 10 power 82. 10 power 82, you can say, is almost zero compared to 10 power 260, because 10 power 260 is so much greater than 10 power 82, which really means that the number of proteins that are possible theoretically is enormously large. Whereas if you take our body, you may find 100,000 proteins in our body, different proteins. Not 100, we will have billions of protein molecules. But if you see, ask how many kinds of proteins are there in our body, it would be about 100,000, which means out of the 10 power 260 proteins, some 100,000 are, like if you take all living organisms, maybe a billion proteins may be present in our biosphere. That is almost zero compared to the number of proteins that are possible in the universe. That is the picture here. I have taken it from one of the David Baker's publication. The blue here shows the possible protein space. The white circles here show the actual proteins formed. This is, of course, a highly exaggerated number. The blue is so much large that it extends, probably, if you want to consider it, you should extend it on both right and left and top and bottom to up to the end of the planetary system. Such a large sequence space we have, but the actual sequences incurred are only a few. This will, lead us to very fundamental questions. Why are these proteins chosen for life? Why not other proteins? Can we construct life with entirely different proteins? You will see that David Baker's research gives partial answer to these questions. Anyway, the sum and substance of this slide is to point out that the sequences possible for proteins is infinitely large, out of which a minuscule number of sequences are used in living organisms, which means when you see in molecular biology, it is often possible to select a protein from our body and change it to have new properties. If you do that, you will be exploring only the area around existing proteins. You won't be exploring the vast regions of space which are not found in living organisms. So the white circles here represent native protein. Vast volumes of blue region are unexplored proteins. But why this white circle is not a just dot because it represents a native protein, but a small circle. Of course, it's highly exaggerated. The circle size should be very, very small. That we can understand. You see, I have just quoted here a sentence from English 500 years ago. What think you of this English? Tell me, I pray you. It is a language that will do you good in England, but passed over, it is worth nothing. But you see that modern English is quite different from this. Why did it happen? Languages evolve. With time, we change. We speak Kannada speaking people. We know how difficult it is to understand, let's say, Pampa or Ranna, because they use Kannada, which is 1,000 years old. So languages evolve. Similarly, our DNA is copied again and again in every generation, in every cell division. DNA copy is made. And there will be copying errors when DNA sequence is copied. So actually, there will be change in the DNA sequence. If there is a change in the DNA sequence, DNA, there will be a change in the protein. For example, here you see I have quoted mouse, human, cow, bovine means cow, and rabbit. This is the protein hemoglobin, which carries oxygen to all our tissues. This hemoglobin is made up of this string of amino acids. The mouse hemoglobin starts with an amino acid called methionine, followed by valine, leucine, serine, glycine, glutamic acid, aspartic acid, etc. See, this is one letter code. I am reading the actual name of the amino acid. You take human, it also starts with methionine, valine, leucine, serine, then there is a change. You see, we differ from mouse in the sixth position. So you see, we are very, very, very close to mouse in terms of our proteins. That tells you that we have evolved from a common ancestor. This is the very powerful proof of Darwinian evolution. But anyway, you see, this picture again is from Ted Baker, and you see, if you take this white region, what people have tried to do is to select, let's say, human hemoglobin, make changes in the amino acid, which is like replacing one of the colored beads with a different colored bead in our string of beads. If you make the change, what kind of property change can happen? For example, if you replace all human amino acids, wherever they are different from mouse, if you change it to mouse amino acid, would it behave like a mouse hemoglobin? Actually, yes, it does indeed. Because of this kind of changes people have made, we explore a protein and the sequences that are close to it but not identical to it. That is why the white circles have some area, which means we have explored a little bit in the surrounding. Just like, our space exploration has not only explored, you see, we have explored what is on Earth, all the islands we know. We also know something about the moon and the Mars, and a little bit less about Jupiter and Saturn. So we have explored a little bit of space. How much have we explored compared to the size of the universe? Of course, we have explored nothing. It becomes less than a mere point. Where our explanation is very small exploration. Same way, what we know about proteins is only a minuscule fraction of all possible proteins in the universe. Anyway, now we have seen that protein sequences have a enormous space. That means the sequence space of proteins is very large. But if you take the structure space, not the sequence, what I mean by structure space, you see this necklace of beads. It is lying with some shape here, S shape. It has an S shape here, you can see. But I could have arranged it in U shape, or A shape, or Z shape. I can give any shape. How many shape can I give to a necklace of 200 beads? That is the same question if you translate to protein. If you have a 200 amino acid protein, how many shapes can this protein assume? I won't be able to present the actual precise technical data on this, but anyway, it turns out that even this number of structures one protein chain can assume. You see, we are not talking about all proteins. You just take one protein of 200 amino acids. How many shapes can it assume? That problem was first recognized by our own Professor G.N. Ramachandran, who is actually the founder of the department where I have worked all my life. He realized that there are two places for every linked amino acid. There's one amino acid here, say one amino acid here in the right hand side picture. Between these two, there are two places where rotations of the structure are possible. If you have half the structure here and half the structure elsewhere, you rotate about this, the right half of the structure can come to different spatial relationship with the first half. See, exactly like, if you think of how many shapes our body can assume, I can bend over my, let's say, knees, or I can change shape of my fingers, so I can give different shapes to my body. You can ask how many shapes a human body can take. G. N. Ramachandran thought, how many shapes can a polypeptide assume? It just tells out that it is infinitely large. Again, infinity is a very complex concept. All that we mean is it is extremely large. So the number of proteins possible is infinitely large. And if you take one protein, the number of structures it can have is infinitely large. It is so large that I have a simple calculation here. You see, even if you assume, let's say, our raised hand has one shape of our body, and a hand down is another shape. Like the two shapes you think of a human body can take. Like that, if you take 9 shapes possible for a link between two peptides, it will be 10 power 95 possibilities for just a protein of 100 amino acids. Put it simply, if you have a protein chain of just 100 amino acids, it can assume one of the 10 power 95 shapes. It's an enormous number of shapes. 10 power 95 is actually 1 followed by 95 zeros. It is such a large number. Let us say the protein, you see, let me actually come back to this slide. I'll go to this slide. Let us say you can, in all physical systems, there is a principle. Anything trying to go to minimum energy conformation. This is not a very difficult concept. If there is a stone on the top of the building, that's an unstable state. If somebody kicks, that will fall to the ground. So whatever is the lowest position, that is the position of lowest potential energy for the body. So it comes down. That is why a stone at the top of the hill or water at the top of the hill will flow down. So it is energy, minimum energy. That is the most comfortable position for any system. The system collapses into minimum energy state. But which is the minimum energy state? If you ask for a protein chain, let us say what is shown in the right hand side, there is a protein chain. If it folds like this, let us say which is on the right hand side, it has a minimum energy. How does it know that it has to fold it to that shape? One way is to test different folds. Fold like this. Is it very comfortable? No. Fold in another way. Is it very comfortable? It can explore each shape one by one. If you do that, the problem becomes a huge problem. For example, you see there are various examples that are given. Let us say we train a chimpanzee to type on a typewriter. It actually types one letter every second. It has been trying to type very fast. It is hitting one button every second. So, but it has no sense. It is only a monkey. So it is typing randomly. So you will get sheets of typed pages which have random combination of letters. How much time will it take for the chimpanzee? Let's say it is typing in Canada. uh, Govinahadu, some famous Kannada song, or if it is English typewriter, how long will it take for him to type Shakespeare's one of the sonnets? Of course, you see, if it's randomly coming, one of the random things will be Shakespearean sonnet. Another way to imagine is that there is a hall with chairs. The hall is infinitely spread on the right and left. One of these chairs is most comfortable. Which chair it is? If you have to do it, you have to actually sit on every chair and find out where you find most comfort. But that will be an unending job. But putting it technically, see, actually, the energy landscape of a protein is a rugged surface like the one shown on the left hand side bottom. Somewhere here, there is a deepest valley. Other valleys are not that deep. The protein has to go to that valley and that shape, which ensures energy minimum. How does it go? You see, if you are stuck in some local valley which is not very deep, you won't see the valley which is deeper somewhere. You will have to just go over the entire region to find the lowest valley. That will take infinite time. In fact, Levinthal proposed that the time taken for the protein to find its shape would be infinite. So no protein can actually fold. But actually proteins fold. That is why we call it Levinthal paradox. In fact, if you estimate how much time it will take for protein to really find its shape, if it searches all possible structures, it will be something like 10 power 10 years. That will be much longer than the lifetime of the universe, which actually, that's why it's called a paradox, that proteins cannot fold at all, but they do fold. So since they fold, there must be some ways in which the proteins do not explore all those possibilities. Something channels them into their minimum energy structure. It is as if there are boards in our Himalayan range which tells you go right, go left, don't turn here, this will lead you to dead end. So you follow the signs, you will actually go to the deepest valley, which means proteins follow some rules, and these rules lead them to the minimum energy conformation. What are these rules? Can we find these rules? That is the uh, one of the most important questions. In fact, in 1963, Unfinson, who got Nobel Prize for his work, showed that if you take a protein, which I have shown you on the right hand side top, a protein which is well folded. You can see it has a very characteristic fold. It has a structure. You heat it, or you can actually treat it with a compound called urea and mercaptoethanol. It loses its structure, and the beads get stretched, and the chain becomes elongated. This is a compact chain. If you think of the necklace with beads, this is a compact structure of those necklace. But you treat it with heat or some other compound, it loses structure, and the bead necklace becomes a long chain. You remove those, you cool it, or you remove those compounds, it actually folds back to its original structure, not any other structure. Unique original structure. It actually surprisingly fraction of it, a small time, which means it is not exploring all possible shapes. There is a folding funnel which directs it. It is exactly like you have signposts in Himalaya which will lead you to, let us say, either the top mountain or the deepest valley. You see, Everest is the top mountain. So everywhere there is a board, this way to Everest, this way to Everest. So although you may start in Himachal Pradesh, you will go to Mount Everest within a reasonable time. You don't get lost in the Himalayan range. That is the idea. What are these signposts? Then that is the question we have to ask. Well, this has been discovered with extreme amount of work. The amount of work done by thousands and thousands of people over half a century. And let us actually look at some of the lessons that have been learned. On the left-hand side here, I have shown a polypeptide chain. And the individual beads here are amino acids which are linked together to make the protein. And this will fold into a unique structure. If you look at more closely at you see, all of these are made up of atoms. Our whole universe is made up of atoms. Any matter that we see is made up of atoms. So these are just the circles here, just write the name of the amino acid. But the amino acid itself has an atomic structure. If you expand that amino acid structure, you will get the structure on the right hand side. This is, of course, a very small piece, which is just 3 beads here taken, and their atoms are expanded. But if you expand all these beads, there will be hundreds of atoms, and our mind has trouble grappling with the complexity. At one end of the polypeptide chain, you see on the left-hand side at the top, you have N and 2 hydrogen atoms. That will be N and with hydrogen atoms here. Of course, it will be NH3 with a positive charge. And then you have a series of carbon, nitrogen, carbon, nitrogen, carbon, nitrogen atoms. At the other end, you have a so-called carboxyl group which carries a negative charge. In between these, you have these branches. There is one branch here, there is another branch here, there is another branch here, there is another branch here. There are branches. Every amino acid has a branch like that. Every one of these beads has a little projection that we call side chain. That actually the sequence of these side chains give the protein its characteristic properties. So you see, actually, if you think of protein, how complex it is, I just want to illustrate to you. See, these are compounds. On the bottom here left, you see a water molecule. It is, all of us know, even a primary school child knows now, water is made up of 1 oxygen and 2 hydrogen atoms. The next to water is shown methane, which has a carbon atom and 4 hydrogens. This is the common gas that we use to cook our food in the kitchen. And The third molecule here is an alcohol. I cannot get rid of this KSTA. Hide. Okay.
Hide button ಗೊತ್ತು ಅಂತ ಹೇಳ್ತಾರೆ because that is obstructing.
No, I will hide. I'm sorry for the interruption. I this
cursor
cursor here. All right, now it is hidden. All right, the molecule here is an alcohol, ethyl alcohol. This is the main constituent of all hard liquors and beer. If you drink it, you will get intoxicated. These are really small molecules. Compared to that, the most common molecule in our body, hemoglobin, that has a structure like this. Each dot in this graph is an atom. Instead of 3 atoms, it has about 6,000 atoms. It is a forest of atoms, and we cannot make out what is what. So we want to represent it in ways in which it is comprehensible. Now, if you put a little ball at every atom, ball of a size which represents atomic size, you will get a picture which is shown on the right hand top. That is also hemoglobin, except that the atoms are replaced by small balls. You can see there is a small hole at the center of the hemoglobin. Hemoglobin atoms are not dots, they have their own body. So if you represent that body, you will get such a picture. But even this is very complicated. What we can see is that there are many red balls. Red balls are water-loving balls. They want to interact with water. Whereas gray balls would rather avoid balls. So we can find the distribution of balls on the surface and inside of the protein from a picture like that. So it is useful. But the most useful would be just to trace the polypeptide chain. How our chain of beads or chain of amino acid is folded in space. And we have, actually, there are 4 chains. Hemoglobin is made up of not 1 chain, but 4 chains. So we say hemoglobin has quaternary structure, which means it is made up of more than 1 chain. The discs that you see here are not, they are a cluster of atoms called heme group. And that heme binds oxygen in our lungs and supplies it to all our body. It is actually, I would call it hemoglobin as the king of proteins. It has been studied very extensively for almost a century because, breathing is the most important aspect of our life. Once breathing starts, we die. So this breathing, the purpose of breathing is to supply oxygen to all parts of our body. And the agent that supplies oxygen is hemoglobin. And the 4 heme groups which bind oxygen are these discs. This is the structure of hemoglobin. It's so complicated. How do we go about learning? Well, actually, you see, you have to have exceptional intuition to understand these molecules and make sense out of these. And I want to give a very simple illustration of how intuition can play such an important role. In England, there was a great lady called Rosalind Franklin. She actually exposed DNA, actually, you should say DNA fibers. For simplicity, I will just say DNA. If you expose any DNA, you won't get it. You have to expose a DNA fiber. But for our purpose in this seminar, we can call DNA. She hit DNA fiber with X-rays and she made a photograph. And this photograph has some information about DNA. What is it, that information hidden in this picture was worked out by 2 people, Crick and Watson, who built a structure of DNA based on this picture. And this was built because of intuition, not because logically they deduced the DNA structure from the pattern. They couldn't, there was not enough information in the photograph to deduce what is the structure of DNA. But their intuition helped them. They built a structure of DNA, and for which both of them were given Nobel Prize. Rosalind Franklin would have been included in the Nobel Prize, but by the time Nobel Prize was declared, she was not alive. But anyway, these 2 people got Nobel Prize, and that is an exceptional intuition. And such intuition has to be there for proteins also, to understand proteins. We will see what constitutes this intuition. In fact, around the time when they built the structure of DNA, Linus Pauling You know, the person you see in this picture is Linus Pauling. He is perhaps the most talented chemist of 20th century. He was thinking about what kind of structures the polypeptide chains can have. To put it in another way, if you have that necklace of beads, in what ways can the bead can fold on itself? You know, that is like asking the question about the structure of DNA. By that time, he himself, you know, Linus Pauling himself, had discovered a great deal about a bond called hydrogen bond. You know, atoms in a molecule are connected by a bond. Otherwise, they will fly away. You know, if they are not bonded in water, oxygen and hydrogen are bonded. So water always has H2O structure. If they are not bonded, hydrogen will fly its way, oxygen will fly its way. So these are called covalent bonds, the bonds connecting the atoms. They are very strong bonds. Even if you put a molecule in boiling water, the covalent bonds do not break. But a much weaker bond, but nevertheless important, is the so-called hydrogen bond. The existence of hydrogen bond itself was discovered by Linus Pauling. You know, in hydrogen bond, if an oxygen is opposite to a nitrogen which is bonded to a hydrogen atom, see there should be a hydrogen atom which is not shown in this picture attached to the nitrogen, and oxygen should come exactly juxtaposed to these. Then there will be a bond between these two called hydrogen bond. And that hydrogen bond is like clipping the two chains. You know, these two chains are clipped at these places, and then it will be a very stable structure. Similarly, if the polypeptide folds in a helical fashion as shown in the left, it goes up as a helix. Then the nitrogen atoms will come exactly above oxygen atoms, and there will be hydrogen bonding. Well, you see, Linus Pauling realized that when a chain is unfolded, that means our beads, necklace is completely unstructured, then all the nitrogen and oxygen atoms will hydrogen bond with water. But when they fold, those hydrogen bonds with water get lost. So they have to be internally formed. Otherwise, the fold will not be stable. It will open up again. So he proposed that the proteins will have this structure or this kind of structure when you discover the structure. This prediction was made much before any protein structure was determined. He called this as beta sheet and he called this as alpha helix. So he said proteins will fold into beta sheets and alpha helices. That was a prophecy, but it is a scientific prophecy, not astrological prophecy. And actually, when structures were done, it was actually found. You see, that was done in the 1950s, Pauling's proposition. But only in the 1960s, people learned to determine structures of proteins. How do they determine the structures of proteins? That is X-ray crystallography. That is the technique they use to do structures. What you have to do is isolate your protein from the body. Let's say rabbit blood or rabbit muscle. You isolate the protein and then you make crystals of the protein. Take the crystals, put it and hit it with an X-ray beam. Then you get a picture which is shown in the left hand bottom, right hand bottom. And you analyze this picture, you can eventually determine the protein structure. This technology was initiated in the 60s and perfected over the next 3 decades. So actually, it was such a phenomenal success. I will come how phenomenal it is in the next slide. You see, if you look at this, I have titled this X-ray is the most powerful method of visualizing the atomic world. You can see atoms with X-ray crystallography. It was the most powerful technique, but I have put a question mark at the end. The reason is now there is another technique which is now competing with X-ray diffraction and taking it over. You see, this happens in science all the time. I was an X-ray crystallographer all my life, which was very good because X-ray crystallography was the most powerful technique for visualizing the world. But now another technique called cryo-electron microscopy has come into vogue. This was developed just over the last 20 years, but it has become already so powerful that it is replacing X-ray diffraction. So today, if you want to work in structural biology, you have to use electron diffraction, electron microscopy. And here is David Baker with one of his molecules, whose structure was determined by the technique of electron microscopy. I have illustrated the technique at the top right of the picture, but we don't have time to go into that. But it is an alternative technique which replaces X-ray diffraction. But as happens in all science, to discover a technology takes time. Once it is developed, once it is discovered, the development becomes very fast. See, 1970, where I started my research career, 70s, there were almost no protein structures. We started working on protein structures in India. No protein structures, almost zero. But I actually 1990, still very few protein structures were there, some of which were actually added from Bangalore. Then you see, I retired in 2015. Then you see the protein, now we have 200,000 protein structures known. That means it's a phenomenal explosion of knowledge. We know the structure of 200,000 proteins. Now, what do these structures mean? Baker and his colleagues analyzed these structures and obtained insights from what is it that is important in protein structures. And then they used those insights to design proteins with very novel functions. That is the rest of the story. Okay, so first, I can only give a bird's eye view of what was discovered. It is a discovery of 30, 40 years, and it is really what has been discovered is remarkable. We first actually focus on the forces that stabilize protein structures, which means, translate it into common man's language, we have necklace of beads. It folds into a structure. Why does it fold into that structure? What forces stabilize the structure and hold it such that it doesn't open up again? Of course, we have already seen covalent interactions, which are chemical bonds between atoms. But then this cannot be broken. This will be there whether the protein is folded or unfolded. So these are not really important for how a protein folds into native structure. The important factors are so-called non-covalent forces. And any a positively charged entity attracts a negatively charged entity. We call it an ionic interaction. It is the attraction between positive and negative charges. There are pairs of positive and negative charges in the bead that we saw in the chain, and they tend to make those two beads come close together. You see, hydrogen bonds in the chain, one bead can hydrogen bond with another bead far away, and that tends to make those two beads come together. Similarly, we have van der Waals interaction. This will be a small attraction between any two pairs of atoms. What does van der Waals interactions do? They make the interior of the protein very dense with atoms. Van der Waals interactions become large when atoms are crowded. If they are not crowded, then these interactions don't exist and it will actually be not that stable. Then I won't go into other interactions, dipole-dipole interactions, stacking of aromatic groups, and entropic forces. I won't go into those details, but David Baker and his colleagues could actually find mathematical expressions to represent these weak forces. You see, in all science, we have a model of investigation. If you have a problem, the problem may be how to, take luggage from India to America. You have a physical problem. You represent it in a mathematical way. So the mathematics should reflect the physical reality. Then solve the mathematics, and the result should be translated back to physical reality, which means the best path or how you navigate in the ocean to reach America. Something like that. David Baker and his colleagues found mathematical expressions to represent all these interactions. Then they could ask questions: how do we maximize these interactions such that we find the most stable protein fold? It is obviously a mind boggling problem, but they could solve. Well, if you look at protein structures that have been solved, you often find some structures, like, for example, you think of those beads. The beads go straight up. On the left hand side, you will see the chain going straight up without any kink. Then it bends and comes down in a helical fashion, and then a kink and goes up again. I already mentioned that the straight structure Linus Pauling called as a beta structure, and this this helical structure is an alpha helix. So this is beta alpha beta. Here is a beta beta beta. Here also is a beta beta. But you see the chains are parallel here, anti-parallel here. Like that, there are some ways in which polypeptides often fold. You see, this is a very nice structure. The polypeptide chain, or our chain of beads, starts like this. There's a king, comes down, turns, king, and then turns and comes like this. There are four chains which are in beta conformation. These structures are very often found. Then we can ask, why are these structures found? What is it that is contributing to their stability? Such similar work should be were done in the last 30, 40 years. And then David Baker and his colleagues could summarize the features that stabilize protein structures. I will just read this slide. I think it will make sense to most of you. I hope it makes sense to most of you. If not, I think you will send me an email, I can answer. Non-polar amino acids, that means amino acids which are hydrophobic, water hating. I told you that some amino acids don't like water. Other amino acids love to be in the presence of water. Non-polar amino acids have to be inside the proteins. Polar residues which love water has to be exposed to water, and if they are not exposed, they should be internally hydrogen bonded. Their hydrogen bonding should be satisfied internally. Isolated charges are rare, are never found in interior of protein. No charge group should be in the interior. But whereas a pair of positive and negative charges sometimes could be in the interior of the protein, they can also be in the exterior. And inside should be very tightly packed. There should not be holes inside the protein. When you wind our bead chain, there should not be a hole inside. You don't wind it around a hollow sphere, for example. You take a hollow sphere and wind your beads around that sphere. That will not be a stable structure. So there should not be large holes. These observations and many, many more will go into design of proteins. And you have to be aware of the whole plethora of energy of proteins, polypeptides, and design polypeptides which are going to be stable, because all these conditions are actually obeyed in your design. That is the challenge of the design. Actually, one of the things which is very easy to convey in a seminar, I have picked up. See, if you have a helical structure, the helix goes round and round itself. It is like a staircase, helical staircase. I am sure all of us have climbed helical staircase. The steps go round and round. But if you look from the top, see, if you look from the edge, it looks like a helix. But if you look from the top, all the steps get projected into a circle. That means a helical structure looks like a circle. And you see the residues that occur along the circle. In a real helix, in some proteins, the residues are distributed such that the yellow amino acids that you see here, each bead is an amino acid, exactly like our chain of beads. There it was a plastic piece, here it's an amino acid. These amino acids all love to be in contact with water. These amino acids don't want water. So this helix is is, we call it antipathic helix. One side of it wants to be with water, another side doesn't want to be with water. Hydrophobic and hydrophilic. This distribution, this is actually from a actual example from a protein. So you do find such helices. How do those such helices, how can we use such helices in designing proteins? See, we can actually make, all right, this slide is not so much in design, I will tell you. When a polypeptide folds, see, first of all, the polypeptide is a string of beads, or of course, the strings are amino acids. That is what is shown in the left-hand side. We call it the primary structure of the protein. But it can, we already have seen, it can fold into a helical structure. This we call secondary structure. It can also make that beta structure where hydrogen bond is between adjacent chains. Then it folds into a compact structure that we call tertiary structure. If units of this kind come together, there are two chains here, we call it quaternary structure. Now, already I a little bit, I should have used some other slide, but doesn't matter. Actually, the title of this slide says, or at the top, if you know the road signs, you will reach your destination. That is the philosophy that David Baker and his colleagues have used. So you have to know the road signs. Well, so using the roads, knowing the road signs, they constructed a program called Rosetta. This will lead you to the structure of the protein if you know the sequence and the road signs. So construct a, see, I think I have only 10 minutes left. I cannot go through the slide for lack of time. See, I started 15 minutes late, so I presume I can take 15 minutes extra. Otherwise, I have to start at 11:30. I will stop at 12. So construct, these are the road signs, let us say. And if you follow these road signs, I could go through, no, I don't think I have time. If you go through these road signs, you will end up with the structure of the protein. In other words, an understanding of how proteins fold and what interactions are important were most essential to develop this Rosetta program. It has been phenomenally successful, and virtually tens of thousands of people have used this program to come up with a possible structure for a given polypeptide fold, which actually translate to, in a simple term, if you had that chain of beads, if you know which colored bead follows which colored bead in the sequence, you can come up with a result which tells you how that chain will get folded in three-dimensional space. In other words, knowing the amino acid sequence, you will be able to find what three-dimensional structure it is likely to assume. This uses the road signs to arrive at, starting from the open chain to come up with the full folded chain. It uses the road signs. The road signs are the rules that protein structures follow in folding into their native structures, such as helices, existence of helices, beta sheets, hydrogen bonding, polar residues out, hydrophobic residues in. How to use these rules to construct protein is not a trivial problem. That is exactly what David Baker's lab achieved. In fact, that protein prediction program, which was developed by them, can be used for dual purpose. You see, to understand all the details of this Rosetta program is beyond the scope of this lecture, naturally. So we won't attempt that, but we will see what kind of use Rosetta has been put. You see, it can be used for prediction. That means, given the sequence, how to find the structure. Translate it to our beads problem. Given the sequence of which colored bead follows which colored bead, we can tell how the chain gets folded in space. So here the problem is, given amino acid sequence, how does it fold in three-dimensional space to give its unique three-dimensional structure? This is the prediction side of Rosetta. But we can also use it for design, which means I want to construct a protein which has a specified structure. For example, you construct a structure, a helix followed by a beta, followed by a helix, and they are arranged in space in a particular fashion, which means we desire a structure. See, I actually fold my beads into a particular structure. I want to choose which colored beads I have to use such that the necklace will have that particular shape. That is the design problem. Given the sequence, given the structure, I want a structure, a helical bundle, for example, a bundle of helices. If I want a bundle of helices, what sequence should I choose? That means if I want my necklace to be a bundle of up and down structure, what colored beads do I use for the necklace? This is a design problem. We have a structure in mind. We want to select a sequence that folds into that structure. That is the design problem. Rosetta can address both the prediction problem and the design problem. There are some more details below, but I don't have time to go into that. But I will show you some of them. See, for example, at one point of time, about 8 years or 10 years ago, they decided to actually design a structure which looks like this. See, which means our necklace should fold like this. You see, the colored beads should fold like this. There is a particular fold here, three-dimensional structure. If it has to fold, what sequence of amino acids should be chosen? That is the reverse question they solved. And they actually made this protein. Proteins can be made because of advances in another field called molecular biology. See, you can synthesize pieces of DNA with specific nucleotide sequence. We call them genes. And those genes can be incorporated in a bacterium such as E. coli, and the E. coli makes the corresponding protein. But that, let's not get into molecular biology. But anyway, they wanted to have the structure shown at the center of this figure between the two. That structure they wanted to have, and they designed it, and which means they found out which amino acids will make what sequence of amino acids go into structure. They made those amino acids, and they solved the structure by X-ray diffraction, which is shown in the right hand side. These two structures are identical. It's absolutely amazing that they designed ab initio a protein structure, and lo and behold, the actual structure designed by them is the structure into which their design sequence folded. So they actually, in the course of last decade, they have developed many design strategies. In other words, if a structure is already known, can we do it with a very different sequence? Or if you want to, the most important here is de novo design. See, if you remember the blue space that we saw in the very first or second slide, the sequence space is unlimited. There are so many sequences, but life has chosen a minuscule fraction of the sequence for its use in living organisms. What about the other sequences? Can we explore those regions which are not found in any organism? This also brings us to an interesting question. If someday we are able to see life forms on other exoplanets, in some other star system, some other galaxy, will they also contain nucleic acid and proteins like we do? Or will they have very different proteins, which are unknown on planet Earth? That actually begs the question, can you devise proteins which are functional but very different from the proteins in our body? That is what de novo means. De novo means not found until now. So sequence unknown, structure unknown. That is, we don't know the sequence, we don't know the structure, but we want to develop a protein with a particular architecture. So you define the architecture. That means you go to an architect and devise a house plan. Can you construct that house? Here, of course, you don't talk about house, you talk about protein architecture. Then you devise both the sequence, and that gives the structure. Well, I am almost come to the end of my time. I will just show you a happy structures that have been designed ab initio. See, it is not just science is never done in isolation. It is not just David Baker that who designed proteins. He got the Nobel Prize because he was at the apex. But there were others who have done very similar work. See, Regan and Degrado devised, de novo made the structure. It's a helical bundle. All right. But this structure was the structure on the right was actually more difficult to devise. But actually, some Chinese workers devised this protein. It is also kind of ab initio. It's not totally ab initio as they speak, but see how well it is internally packed with these rings at the center. Like that, many. And Baker himself devised the structure and experimentally found structure is identical to this. I have shown here again the internal packing and even the branches or side chains are exactly in the way it was supposed to be. The design showed these residues will actually look like that, and experimentally they actually looked like this. That means whatever they designed was very precise. In other words, the building constructed was exactly like the plan in the paper. In other words, what they designed in their lab, the actual physical protein that was produced had identical structure, which means really we can now make proteins which never exist, which do not exist in any living organism on earth. We can create novel proteins. Now I will actually just go through, some what I call ab initio design galore, which means these structures are divided absolutely ab initio. There is no protein like this in any living organism. They are new proteins. They belong to the region in the blue map which I showed outside of the white circles. These are structures that are possible for proteins. So if you go to an exoplanet, maybe the organisms there may have this kind of proteins. Of course, they might not have proteins at all. We don't know. But these are proteins that are possible which do not exist in living organisms. So you see, there are other design principles, how to use a motif to make symmetric structures. This also they have explored in great detail that has allowed them. You see here, there is a six-fold symmetry. No, I think 1, 2, 3, 4, 5, six-fold symmetry, eight-fold symmetry, ten-fold symmetry. They have very designed symmetric structure. They have also like this carved knife-like structures. All these are designed which are not present in living organisms. I will skip that. You see, I cannot tell you what parametric backbone generation. This is another technique for de novo design. They have used this principle also to design helical bundles, basically. See, there is a three helical bundle. Here, there is a three helical bundle and a four helical bundle. They have designed five helical bundle and three helical bundle. This kind of bundle structures also they have designed. These are based on some other principle called parametric backbone generation, but which as I said, is beyond the scope of what we can cover. But see, they can design this kind of closed structure shown at the top, made up of just helices. What is the use of this? I will tell you what the use of that as a last part of my seminar. See, these are all helical bundle structures. Well, you see how beautiful these design structures are. All these are de novo designed. And I think I will probably end with this. This is the so-called icosahedron. This is the shape assumed by many viruses. And they have designed this. Virus is naturally occurring, but these proteins are designed to take icosahedral shape. These can be made into vaccines. How it is made is another part, another whole story, which leads another seminar. But these can be vaccine candidates, first of all. A little bit of such work has been done by Savitri in her laboratory also, using natural viruses. But these are completely ab initio designed structures, which also can be used as vaccine candidates, or they can be used to deliver drugs to specific parts of our body, drug delivery systems. both of which are very important in today's pharmaceutical industry. See, there is more of such structures. All these are design structures which do not occur. See, there are more design principles and more structural designs, the so-called, I said parametric, here disulfide linked, cross-linked, hyperstable mini proteins. Well, let me conclude it by saying David Baker has designed a lot of proteins, but you ask why design? First of all, it gives us fundamental understanding of how proteins work, and how proteins work decides how our health stays all right, how we behave, everything. Whatever we do depends on proteins. They provide vaccine candidates. They can be made into virus inhibitors. We can design enzymes with specific functions. For example, enzymes that can actually create clean environment by digesting discarded raw materials. We can make new enzymatic functions which can be used for pharmaceutical applications. We can actually use inhibitors of bacteria. We can target drugs to any part of the body and many more. It is really your ingenuity and originality that allows you to make this field applicable to many industries, both chemical industry and pharmaceutical industry. Because of these fantastic achievements, David Baker and his colleagues were recognized by the award of Nobel Prize for him. I had a little bit on what is the future of this field, but I have run out of my time. But I think I have made some sense, and I am very glad that students have attended this seminar, and I thank you for attending this. And finally, I thank the Academy for giving me this opportunity to present some glimpses of this great piece of work. Thank you very much.
Sir, thank you very much for an excellent exposition of Nobel Prize research in chemistry. I'm sure all those who attended the webinar will be highly benefited by your talk, sir. You started with basics and history, then complexity of structure, then Dr. G.N. Ramachandra's research, then hemoglobin molecule, Dr. Rosalind Franklin's work, and Crick and Watson's work, then features of protein structures and structures. You also at the end gave pics of structures of molecules. Sure, this is an excellent talk, which many will be appreciating, sir, on behalf of KSTA and on behalf of all the attendees who have joined on Cisco WebEx and YouTube platform. I'm immensely thankful for your time and excellent presentation. We have with us.
Finally, you can see me.
Sir?
Can you see me?
Yes, sir. Yes, sir. We can see.
Fantastic. At least the audience know who is presenting. I wish I had this. All my dance and drama were not seen during this presentation. I wish I had this at the beginning, but we had some problems, so it's all right. Do you allow questions?
Yeah, yes, sir. Yes,
good.
sir.
Good,
sir,
good. Now you can ask.
I am also thankful to Savitri madam for helping you to deliver the.
Otherwise, I wouldn't have been able to give this seminar. I don't know why this problem. No time I have encountered this problem. All of a sudden, something happened. I couldn't log in at all. But anyway, finally it got resolved. So it's fine.
Sir, who is the director at BLD Medical University. Sir, over to you for your.
Yeah. Thank you, sir. Thank you very much, Dr. Ramesh, sir. And thank you, Dr. Murthy, Professor Murthy. Sir, you have really on the heart of everybody here, discussing about the fundamental and basic of the chemistry, which is required to understand the protein in a better way. And I belong to a medical sciences. I'm a medical doctor and a scientist. So in my background in a medical college is not exactly the way your expertization and your deep knowledge in this field and your own work, obviously. But what I understood very clearly that this protein chemistry, what we are being taught during our biochemistry, only 1 year teaching in the medical, is not sufficient to understand the deep cause of various disease and root cause and possible way to cure it. So you have enlightened that part by the protein chemistry, given a different shape, that how it actually changed. Basically, I'm interested to see that foldings. So how the foldings actually take place and make it difference, you have so beautifully explained. It will really make the changes and the mindset of the young generation to understand the subject core, to get the deep knowledge. So thank you very much. My only one question and query and doubt also, that is that this protein, if we able to predict and we able to make it, what will be the future? Because you have said in the last set that you have not discussed about the future. Future may be scope, but future may be disastrous also by developing certain type of coding and making some twist in the folding and make it different, which can be reactive to certain type of organs more specifically, or it may cure also, it may change it into the better shape. So we are not sure at the moment. So what you think about it? Just one line, sir.
You see, this is design can be used as I said at the end, vaccine candidates or drug targeting the drug. So the application is really will be immense. There is no doubt that it is going to revolutionize pharmaceuticals, particularly vaccines. So we you see already they have a successful vaccine here, but of course it has to be approved by FDA. But what you can do is you can design a protein which carries an epitope of virus specific epitope. Then it will be a vaccine candidate for the virus. So there are possibilities, and certainly it will be exploited. It will revolutionize many aspects of our everyday life. And also you see proteins. See, if you can produce alcohol, that I mean, if you can use fuel for cars using sugar cane very efficiently, which has been done in Brazil, but this design will allow you to create enzymes which are so efficient that not a trace of what can be converted to alcohol is left. And of course, finally, if you can change cellulose to edible forms, it solves the food problem in the world. Population becomes no bar. Then any population can be supported if water is available, of course. But anyway, you see, we have no way of converting cellulose to an edible form. If that can be done with an artificial enzyme, see, once you can construct an enzyme, you have a handle on what function you want to engineer. So your chemistry knowledge should allow you to construct these enzymes. And already people have constructed enzymes. Brazil has been very successful. In Brazil now it is compulsory that you use alcohol in your car. You cannot use petrol. There are cars which use 100% alcohol, but at least 50% petrol, 50% alcohol you have to use. That will reduce the cost of fossil fuels, and we can depend on renewable sources. So, like that, applications are unlimited. That's all what I can say at the moment.
Yeah. Thank you, sir. Thank you very much.
Thank you very much, sir. Kushal sir and Professor Murthy, sir. Umesh Gatke, questions?
There is only one question from Dr. Jyotirmay Ghosh. How to predict the structure for the post-transitionally modified proteins?
Oh, the same techniques. The same techniques. You see, post-translational modification will perturb the structure to an extent. It probably won't completely alter the structure, but the program used to that you can see. For example, trypsinogen is converted to trypsin as a post-translational modification, but the core of the protein has the same structure. But the additional segment that is cleaved off, that can be predicted with the program. In fact, actually, there is another program called AlphaFold 2, on which there was a presentation yesterday by my colleague Nagasuma Chandra. I was teaching at the time, so I couldn't attend. But she would have told you how that works. That can be used for any protein, even proteins which have been post-translationally modified are modified. If you introduce a mutation, a deletion mutant or addition mutant or substitution mutant, how will it disturb the structure? Even that now can be theoretically investigated. Earlier, only experimentally, you had to create the mutation, determine the structure by X-ray diffraction or whatever technique, and then see what is the effect of mutation. That was done. Effect of mutation was extensively studied in the case of lysozyme using X-ray crystallography. That was done for years with an enormous cost. Now you can do all that with a program, software. So post-translational modification, prediction of the protein structure, what alteration could have happened, is not a very challenging job anymore. You can do it with AlphaFold too.
Thank you very much, sir. Once
Yes.
again, on behalf of KSTA, we are immensely grateful to you for an excellent scholarly talk. We'll keep organizing webinars like this. We'll be inviting you for future.
Yes, you should invite me more often. That will be my pleasure. My complaint is that you don't see, particularly for school children, you make as a presentation. I love to talk to school children more than college because that is the foundation. Or even middle school children, if you can make arrangement in schools and broadcast it. You see, there was a time when our space program was broadcasting special programs for schools, but that I don't know, somehow it was not very successful. But as an academy, we can organize lectures for school students after the school hours, and some people will attend. Even if 100 people attend in the whole of Karnataka, we would have reached those 100 people. If you don't do it, you don't reach those 100 also. So if you arrange, it will be my great pleasure to participate. Or you ask me to go to any corner of Karnataka and combine schools. You see, Karnataka, KRVP used to do such programs. Now KRVP is essentially dead, which makes me very sad. But I think academy should take up the same work. That's my opinion. And all of you have to do it. And we are only servants to you. If you do it, we will be very happy servants. All right. Thank you very much.
Grateful to you, sir. I'm also thankful to Professor H.S. Savitri Madam for making this talk happen. Because of
Yes.
we got this.
Thank you. Bye-bye.
Thank you, madam. And
Thank you, Ramesh. Bye.
Professor Kushal Das, sir, thank you very much. You have always been supporting us. Grateful to you. We have been joined by Dr. Sindhu, Dr. Qureshi, and Dr. Jyotirmay Ghosh. For all of you, big thanks, and also I am thankful to all the audience who have joined both on Cisco Wex and YouTube platform. And we are grateful to you. Tomorrow's talk, Mr. Umesh will be telling it. Umesh, hello.
Tomorrow we have a session on economics.
On economics by renowned economist, Agriculture University. I request all of you to kindly join at 11:30. Thank you.
Thank you, sir. Thank you very much.